Hi, my name is Matthew, and I will be discussing the anatomy and enzymology of fat absorption. So first, I would like to just provide a little overview discussing the route of travel of fat through the body. According to the Silverthorne textbook, nearly 90% of our fat intake comes from triglycerides. Other forms of fats include cholesterol, phospholipids, and long-chain fatty acids. In order for fats to be absorbed by the digestive system, they need to be broken down into smaller lipid droplets. The course of fat through the body starts in the mouth, as can be seen here with this man eating an apple, where in the mouth mechanical digestion begins by chewing. Some short-chain lipids are also broken down at this point by the enzyme lingual lipase. The bolus then travels down the esophagus, as indicated by these arrows. It enters the stomach, where it is churned and mixed with acid to become chyme. It then enters the duodenum of the small intestine, where the majority of digestion and absorption occurs. As we can see zoomed in here to the small intestine, we can see the lumen of the small intestine, specifically the villi, and an enterocyte where most of the lipids are absorbed. So the main issue with which the body is faced in digesting and absorbing fats or lipids is the issue of solubility. Fats are very hydrophobic molecules, and as a result, they are not very soluble in the aqueous environment of the intestine. To combat this issue, the digestive system takes advantage of the amphipathic properties of bile salts. Bile salts have both hydrophobic and hydrophilic regions, as can be seen here in this illustration. And this creates an emulsion of small fatty droplets, which are now soluble in water. Now that these emulsified fat droplets are water soluble, they can start to be broken down by enzymatic digestion. And this is done by lipase enzymes, which will be discussed in this slide. Lipases are synthesized by the pancreas, and they are released into the small intestine along with bile from the liver via the sphincter of Oddi. Lipase, along with the help of colipase, hydrolyzes triglycerides, forming one monoglyceride and two free fatty acids. This breakdown process can be seen in this reaction diagram below. Byproducts of enzymatic digestion, including fatty acids, bile salts, monoglycerides, phospholipids, and cholesterol, are all bundled and stored in micelles, which are a packet of lipid molecules suspended in the aqueous interstitium. The micelles help transport the triglyceride components closer to the absorptive epithelial cells lining the small intestine, which we saw in the first slide. So the process of fat absorption itself occurs as micelles near the epithelial villi. They start to free their lipid passengers, and since lipids are hydrophobic, monoglycerides and free fatty acids are able to diffuse freely across the enterocyte membrane. Cholesterol requires energy-dependent transporters, but also enters the apical membrane of the intestinal cell. So we can see here in this illustration, on the apical side of the intestinal cell, the free fatty acids and the monoglyceride diffusing freely, the cholesterol being transported, and reforming triglycerides inside of the intestinal cell. Once inside the cell, absorbed monoglycerides, free fatty acids, and cholesterol recombine, as I just mentioned, to form triglycerides and are ready to be taken up and used by the body. So the last process, after fat has been absorbed, involves its transport so that it can be used by cells in the body. Once triglycerides are reformed inside of the enterocyte, as we just saw, they accumulate cholesterol and proteins to form large lipoprotein transport molecules called chylomicrons. Like micelles, chylomicrons are amphipathic, but due to their large size, as you can see here, they must exit the cell via exocytosis. After exiting the cell, chylomicrons are still too large to pass through the tiny fenestrations and capillaries to enter circulation. Instead, they are absorbed into lymphatic vessels called lacteals. Chylomicrons enter lymphatic circulation and drain into the venous blood supply just before the blood enters the heart, where it can be pumped to the rest of the body and used by body cells. And last, I have some of the sources listed that help me with researching the anatomy and enzymology of fat absorption. Thank you.